Hello everyone. This class is for nursing students, especially for third semester, and the subject is pharmacology. First unit, Introduction to Pharmacology. So let's see the introduction. So here in this unit, we will see what is pharmacology, from where it is derived, the word pharmacology, and what is the meaning of that, and different branches, everything we'll study in this unit. Okay, let's start. The word pharmacology is derived from Greek word pharmakon, means drug, and logos means knowledge or study. This is a Greek word, the pharmakon, and this pharmakon means drug and logos means knowledge or study. So literally we are studying about the drugs here, that is pharmacology. Understanding how a drug or a substance is likely to modify the living system in the crux of pharmacology. So here when we give a drug, that is any kind of drug, how the living system is getting modified, that we study here. By definition, pharmacology is the science that deals with the study of drugs and their interaction with the living system. So here detailedly we are studying about the drugs and how it is interacting with the system. So why we are taking the drugs? So when we have some illness, maybe to subside some of the symptoms or maybe to cure the disease, we are taking the drugs. So here how these drugs are acting on our systems, various systems, those things we are studying here in the pharmacology subject. Some of the definitions they have given. So here pharmacology is majorly divided into two branches, that is pharmacokinetics and pharmacodynamics. Pharmacokinetics, it is the study of movement of drugs in the body, including the processes of absorption, distribution, metabolism and elimination. So here it tells us about how body treats a drug molecule till it is removed. So for example, we have taken a tablet. So in which route we have taken, where it is going, where it is getting absorbed, where it is getting metabolized, and where it is excreting, everything we study under this pharmacokinetics. Then it tells us about how body treats a drug molecule. So how our body is treating that particular drug molecule, okay, that is pharmacokinetics. Next, pharmacodynamics, it is the study of how a drug acts on a living organism, including the pharmacologic response and the duration and magnitude of response. Pharmacodynamics is nothing but when we take the drug, what changes it is happening in our body. So for fever, I have taken a tablet, where it is acting, what it is doing, how it is changing, okay, that we study under pharmacodynamics. And also here the duration, for how long it is acting on our system. Then magnitude of the response, so how it is responding, everything we will study under pharmacodynamics. Next, we will see the nature and sources of drugs. So drugs are obtained from various sources such as microbes, plants, animals, minerals and synthetics. So from various forms we are preparing the drugs. Preparing is nothing but the manufacturing process. Okay, so how it is? So first one is natural sources, naturally. So here under natural we have many things. First one is plants as a source of drugs. Many drugs are obtained from plants. The active principal drugs are found in different parts of the plant. The drugs are extracted from the particular part of the plant. So here from the plant origin we are making many drugs, and when we are making this we will use a particular part Okay, for the different drugs, maybe different parts. Okay, so particular, maybe we are taking the bark, or we are taking the root, or maybe we are taking the leaves, or maybe we are taking the fruits, or maybe we are taking the seeds. We'll study further. Okay, so here they have given some of the drugs, that is belladonna, we are taking part of the plant is root. Then cinchona, we are taking the bark. Then digitalis, we are taking the leaves. Senna, we are taking fruit. Strophanthus or castor oil, we are taking the seed. You already know this, we are using it. So you may think, for what we are using castor oil? In olden days, to cleanse the digestive system, our ancestors, they used to take 1 spoon of castor oil. Okay, nowadays we are not taking. Then opium. So here seed pods we are using. Then Physostigmine caliber, seed we are using. They have given 7 examples. So here you can write at least 2. That is enough. Next, second one, microbiological. Certain antibiotics are obtained from different types of microorganisms. So for example, penicillin, it is obtained from Penicillium notatum. Then streptomycin drug, the source is Streptomyces griseus. Then the drug bacitracin, obtained from source Mycobacterium bovis. Then drug BCG vaccine, it is available from the source Mycobacterium bovis. So here the microbiological, it is some of the drugs are obtained from the microorganisms. These are the microorganisms and these are the drugs we are preparing from that particular microorganism. Here also you can write any 2, that is enough. Then next, animal as a source of drugs. Plant we saw, microorganisms we saw, third one is animal. Many drugs are obtained from animal sources till today. Extensive research is going on to produce similar molecules in laboratories, but it is not possible for all the drugs. For example, drugs such as adrenocorticotropic hormone, that is ACTH hormone, liver extract, cod liver oil, heparin, anti-rabies vaccine, these and all, the source of drug is animal source. Okay, so still the research is going on, but many drugs are not available from the animal source. Very less drugs are available compared to plant source. That's what they are telling. Next, minerals. Many minerals are used as drugs which are required in small amounts. Minerals we require in very small amount. So here let's see what are the minerals. Example, iron, zinc, calcium, etc. Then synthetic. Most drugs are now synthesized in laboratories using chemical methods. So here synthetic is nothing but it is not natural, it is synthesized in the lab by using the chemicals. They are preparing these drugs by the chemicals. Those are called synthetic drugs or synthesized drugs. Okay. Next, example here for synthetic drugs: sulfonamide, mepacrine, aspirin, neostigmine. But this synthetic source is the very important source. Why? In allopathy we have natural drugs are very less, unlike Ayurvedic and other things. Here we use most of the drugs from the synthetic source. Okay, keep this in mind. Next, human source. Some drugs are obtained from man. Actually, you know, I guess you know this. Example, immunoglobulins from the blood, growth hormone from the anterior pituitary and human chorionic gonadotropins from the urine of the pregnant woman. So here it is prepared from the human source. Okay, if you don't remember anything also, at least you can write immunoglobulins from the blood and growth hormone. Okay, these two you can remember. Next, other sources. Other sources, many biological agents which are proteins in nature are produced by using many techniques in cellular biology, by manipulating genes in microbes or cell culture system. The commonly used techniques include recombinant DNA technology and hybridoma technique. So by using this recombinant DNA technology and hybridoma technology, they are preparing many drugs. Okay, so this is nothing but they are manipulating the genes which is present in the microorganism and they are preparing the drug. Okay, that's all. They are manipulating the gene present in the microorganism. So here, and also they do cell culture. Okay, cell culture, many drugs are obtained, that is urokinase from cultured human kidney. By culturing the human kidney cells, they obtain the urokinase. Okay, then recombinant DNA technology, here example human insulin they are preparing. Then hybridoma technique, so here monoclonal antibodies are prepared with the hybridoma technique. So these are the sources of the drugs. Okay, so hereby sources of drugs finished. It's very easy: natural, synthetic and human. Then next, let's see dosage forms and routes of drug administration. So first let's see the dosage forms. Okay, so here we have solids. So under that we have extract. It is obtained through dissolving drug in alcohol or water and allowing the solution to evaporate. Residue is called the extract. So here when they are preparing, okay, it is dissolved in the alcohol. Okay, and some drugs are dissolved in the alcohol, some drugs are dissolved in the water. Okay, and the solution, it is allowed to evaporate. Okay, it is when we are boiling it, automatically it will evaporate after reaching a particular boiling point. You know that. So the residue which is remaining, so that is called the extract. So that comes under extract. Next, second one is pills. It means a tablet and is made by rolling and binding the drug into sphere. So whatever we are seeing in our daily life, no, the round one, so that is pill. Here it means a tablet. It is made by rolling it, okay, and binding the drug. It is binded togetherly. So we swallow that after going inside, so it opens up and the drug is absorbed. Okay, it will not stay like that only. Okay, it will dissolve. Then powder. So in the powder form also some of the drugs are available. So here it will be very fine powder. Then suppository. It liquefies at body temperature. So suppository, it looks like a triangle shape but little lengthier than the absolute triangle. So here when we insert that, it liquefies automatically due to our body temperature. It will melt. Liquefy means nothing but it is getting melted, okay, like a ice cream when inserted into orifice and produces local and systemic effect. So wherever we are inserting, there it is getting liquefied and it is getting absorbed and it is producing the systemic effect at that particular place. Then ointment. These are the drugs that is semi-solid. It is not solid, it is not too liquid, it is like ointment. So for that semi-solid we call it as ointment. So here why it is semi-solid? Because it has a fatty base. That's why it is not completely liquid. It is like a gel. Okay. Next one is capsule. Drugs contained in the cylindrical gelatin containers. So here capsule. Capsule means inside small molecule, the drugs will be there, but we keep the drug inside one small capsule. So here what happens, when we swallow the capsule, it goes to the stomach and there it melts, then the drug comes out, then the drug is getting absorbed. Okay. Then lozenge, a form of drug which is flavored and released drug slowly when held in mouth. Lozenges are nothing but what the tablets we keep under the tongue or in our cheek, that is lozenges, because when we keep that, slowly it melts and releases the flavor. Okay. Then tablet, powdered form of drugs which is compressed into small disc, same like a paracetamol. Okay. So here actually the drug is powdered form, but it is compressed and made like a disc form. Perfect example is paracetamol tablet. Okay. It looks like a disc. Next, let's see the liquids. Okay. First one is tincture. It is the diluted alcoholic extract of the drug. It is diluted in the alcohol and the taken extract, that is tincture. Okay, but it is in the liquid form. There we saw the solid form, here we saw the liquid form. Okay. Then second one is syrup, concentrated sugar solution containing drug. See here why when we drink any syrup it is sweet, because it is a sugar syrup. Okay, but along with that we have mixed the drug. Okay. Elixir, drug solution made with sugar, alcohol and pleasant smelling substance. Here sugar is there, alcohol is there, plus pleasant smelling agent is there. Okay. Next, suspension, fine particles of a drug suspended in a liquid. See here there is a difference between syrup and suspension. Okay. In the suspension, fine, very fine particles of a drug is there, but it is suspended in the liquid. That's why they have written on the bottle, shake well before use. Why? Because these suspended particles, these are getting settled at the bottom of the bottle. That's why every time when you are drinking the liquefied drugs, please shake it before the use. Okay. Next, emulsion. It is the solution made by suspension of unmixed fats or oils in the water. So here we are using the solution which is made by the suspension of unmixed fats. There the suspended drug, okay, in a liquid, but here it is a suspension of unmixed fats or oils. Okay, and where we are doing it in water. Okay, that is emulsion. Next one is lotion, liquid suspension of medication which is applied to the skin. So it is applied externally. Okay, it is a liquid suspension, but it consists of medication, but it is applied only externally, not taken internally. That is called lotion. So this is enough for today. In the next class, I will continue with roots of drug administration. So let me tell you which book I am referring. This is textbook of Pharmacology and Pathology for Nurses, volume 1, that is for 3rd semester, and volume 2 is for 4th semester. Okay, and this is according to the INC syllabus for BSc Nursing, according to the revised INC syllabus. And the author name is Suresh Sharma. I am using 2nd edition. Okay, you can use the same book and follow my classes. And if you want PPT, please comment below and text me. I will send you the PPT. Thank you. Study well. All the best for your exams.